Hello students, let us start with the next chapter of 11th standard, that is the 9th chapter, Biomolecules. So first we need to understand what are biomolecules. Biomolecules are basically substances which are found in our body. So now these biomolecules, they can be organic or inorganic depending on the chemical composition. So now the question is, how do we analyze this chemical composition? So to analyze this chemical composition, you take a living tissue. It might be a vegetable or a piece of liver or some other organ, and you grind it using a acid. Generally, we use trichloroacetic acid. Then, after you get a slurry, you strain the slurry through a cloth. It can be a cheesecloth or a cotton cloth, and we get two fractions. So these two fractions are the filtrate and the retentate. The filtrate is acid soluble. It contains the biomicromolecules. That is, their molecular weight will be less than 1000 Daltons, right? So this is generally the cytoplasmic composition in the cell. In the last chapter, if you remember, we learned about all the organelles, and cytoplasm was the medium in which all the organelles were suspended. The second one is the acid insoluble fraction. So it contains biomolecules which weigh greater than 1000 Daltons. So we call them biomacromolecules. Macro is for greater than 1000 and micro is for the ones who weigh less than 1000. Right? So this will have molecules from cytoplasm as well as different organelles which are present in the cell. So now we also need to understand how to do analysis of inorganic compounds in the living organism. In order to do that, we weigh the tissue, the living tissue, we dry it to evaporate the water, and the remaining dry weight is burned to oxidize all the carbon compound to gaseous forms. So ultimately we will have CO2 and water vapor. The remaining ash contains the inorganic elements like calcium, magnesium, etc., and some inorganic compounds like sulfate, phosphate. So if we compare the elements in living and non-living matter, then for the Earth's crust, the chemical composition of hydrogen, the percentage weight of hydrogen, carbon, oxygen, nitrogen will be very different as compared to human body. You can see that carbon, which is present only 0.03 in the Earth's crust, is present in a higher amount in human body, whereas nitrogen is very little here, but here it is 3.3 because it forms a part of your proteins and amino acids. Silicon is higher, 27.7, but here it is almost negligible. So now let us study about the biomicromolecules. As we learned that these are biomolecules that have molecular weight less than 1000 Dalton. The molecular weight of biomicromolecules found in the acid-soluble pool ranges from 18 to 800 Daltons. So these include amino acid, lipid, sugar, and nitrogen bases. First, we will learn about the amino acids. So as the name tells you, amino acid, that means it has an amino group, NH2 group, and an acidic group, that is COOH. Then there is a hydrogen, and this R group represents the variable group. That means it can be changed depending on the composition of the amino acid. The amino group and the acid are attached to the same carbon, the alpha carbon. Therefore, they are called alpha amino acids. So depending on this R group, which I told you about, if it is hydrogen, then it can be glycine, CH3, then alanine, CH2OH, then we call it as serine. The amino acids are generally of three types: acidic, basic, and neutral. An example of acidic would be glutamic acid, basic would be lysine, and neutral will be valine. Some of them can be aromatic, that is, they have a ring structure. These include the tyrosine, phenylalanine, and tryptophan. There are total 20 amino acids that take part in the synthesis of protein in the human body. So as we can see here, we can classify them as non-polar and polar. and then the ones with the positive charge, and then the ones with the negative charge. So these are the different amino acids which are found in our body. Glycine, alanine, valine, cysteine, proline, leucine, isoleucine, methionine, tryptophan, phenylalanine are all non-polar. Then we have polar amino acids, that is serine, threonine, tyrosine, asparagine, glutamine, right? Aspartic acid, glutamic acid will have negative charge. Lysine, arginine, histidine will have a positive charge. Another classification includes that whether they are essential or not. Essential means they cannot synthesize by your body. So because your body is not synthesizing them, obviously you have to take it from an outside source. So these include lysine, leucine, isoleucine, tryptophan, and some other amino acids. Then we have the non-essential one. Non-essential means your body can synthesize them. So these include glycine, alanine, serine, and arginine. So these are all the amino acids which are essential and non-essential. Essential means this must be taken from the diet because your body is not making them, right? And then we have all these which are non-essential. That means you don't need to take it from your diet because your body is already making them. Now, if we talk about the nature of amino acids, then they have an ionizable nature. So, the structure of amino acids changes in solutions of different pH. That is the meaning, right? You can see here the Zwitter ionic form is found. You can see how hydrogen is removed, then it acquires a negative charge, and then this can be reversed again. The positive charge of the amino group goes away, right? So this will change depending on the pH of the solution. Next, let us study about lipids. Lipids are obviously water insoluble. They cannot dissolve with water. The chemical composition is CHO, but the number of oxygen atom over here is less. So examples of lipid would be oil, butter. All these are examples of lipids. We can classify the lipids into simple, compound, and derived lipids. Let us first look at simple lipids. Simple lipids are formed of fatty acids and alcohol like glycerol. So the fatty acids are organic acid with a hydrocarbon chain, that is the R group, and it generally ends with COOH. So finally if you see, it will be something like R and COOH. You can see over here. This is the R group and this is the COOH. Palmitic acid, for example, has 16 carbons, right? And then the carbon composition of arachidonic acid is that it has 20 carbons. But you can see that here also there is an R group and here we have got the COOH. The types of fatty acids can vary. They can be saturated or unsaturated. Saturated means there is no double or triple bonding between two carbons. As you can see here, stearic acid is a saturated fatty acid. There is no double or triple bond shown anywhere. It is all single bonds. Whereas unsaturated fatty acids are the ones which have the double bonds. You can see here oleic acid is an example. The double bond is present here. So this fatty acid, it joins with the glycerol to form the fat or the oil. The bonding is ester bond. It can be monoglyceride, that means one glycerol, diglyceride 2, or triglyceride, that is 3 glycerol molecule. So this is how they will look like. Monoglyceride, that means one fatty acid. Diglyceride is 2 and triglyceride will be 3 fatty acid. R1, R2, R3 over here are the fatty acids. The next type that we need to learn is compound lipids. Compound lipids are esters of fatty acids and alcohol with additional group. So you see some other group will be present here. For example, in phospholipid, you have fatty acid plus glycerol and plus an extra group which is phosphate group which is present. So this is the phosphate group. They are found mainly in the cell membranes. The third type is the derived lipids. These are generally products of hydrolysis of simple lipids and compound lipids. So this generally includes sterols like cholesterol. Fats will generally have a higher melting point and oils will have a lower melting point. Now let us study about sugars or carbohydrates. Sugars, as we all know, are sweet and water soluble. The composition is same, that is CHO. The ratio is 1:2:1. So these are the examples of sugar: glucose, which is C6H12O6, ribose, which is C5H10 and O5. This is the structural composition. Then we come to the next group, which is the nitrogen bases. These are nitrogen containing cyclic compounds which are found in our nucleic acid. So we can classify these nitrogen bases into purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, thymine and uracil. Now what are these purines, pyrimidines, and why is it that we put adenine and guanine in purines and cytosine, thymine and uracil? So purines are nitrogen bases with two rings, right? So adenine, guanine have two rings, so they are purines. Pyrimidines are the ones with one single ring. So cytosine, uracil, thymine is having one ring, so we classify them as pyrimidines. Now you need to understand that ATGC, that is adenine, thymine, guanine and cytosine, are found in DNA. Whereas when it comes to the composition of RNA, that is ribonucleic acid, we replace the T with U. So in RNA, you will have adenine, guanine, uracil and cytosine. So now that we know about nitrogen bases, let us understand what is a nucleoside. So nucleoside is nothing but a combination of a nitrogen base and a sugar. So all these nitrogen bases that we looked at, adenine, guanine, cytosine, thymine, uracil, they will combine with a sugar molecule to give you a nucleoside. So you have adenosine, guanosine, cytidine, thymidine and uridine. This is what the structure will look like. Now, when this nitrogen base and the sugar and the phosphate combines together, then you have something called as the nucleotide. These are generally heterocyclic compounds. This is basically the RNA, DNA, that is deoxyribonucleic acid and ribonucleic acid, which is found in your body, right? So what is the nucleotide? The nucleotide is adenylate acid, guanylyl acid, cytidylate acid, thymidylate acid and uridylic acid. This is what you finally have. Now let us learn about biomacromolecules. So as we learned earlier, macro means their weight is more than 1000 daltons. So these will include proteins, polysaccharides and nucleic acids. If we talk about lipids, then the molecular weight of lipids does not exceed 800 daltons, but it forms an insoluble fraction. This is because the lipids are arranged into structures like cell membranes and other membranes. So when we grind a tissue, these membranes are broken up and they form vesicles which are not water soluble. They are hydrophobic. So lipids are not strictly macromolecules. Let us look at the average composition of cells. So this includes water, which is 70 to 90%, proteins, which are 10 to 15%, carbohydrates, which are about 3%, lipids 2%, nucleic acid 5 to 7%, and then we have ions, which are 1%. First, let us start with the proteins. Proteins are a heteropolymer of amino acids. That means various amino acids, they join together to make up the protein. So they are called polypeptides. Why are they called polypeptides? Because they show peptide bonding. As you see here, there is a bonding of CONH. If you remember, what did we study in amino acids? The amino acid had an amino group and an acidic group, right? So these are bonding together to give you a peptide bond. This occurs by dehydration. That is, one water molecule is removed during this process. So what are the functions of these proteins? Proteins are generally used for growth and tissue repair. They are used for transporting things across the cell membranes. Like for example, GLUT4 enables the transport of glucose into the cell. They act as intercellular ground substances like for example collagen. They act as antibodies which help you to fight against various diseases. They act as receptors. You have receptors of smell, taste. Hormones are proteins. Like for example, insulin is a protein hormone. Enzymes can also be protein. Enzymes are all proteins in nature. Trypsin, for example. They can be certain pigments also. Hemoglobin, for example. Hemoglobin helps us to transport oxygen all throughout our body. Most abundant protein found in the animal world is collagen. And the one which is found in the whole biosphere is rubisco. Rubisco is ribulose biphosphate carboxylase oxygenase. Now these proteins, they are folded into various levels. So based on this, we have primary, secondary, tertiary and quaternary structure of the protein. Let us look at the primary structure. Primary structure is a very simple and basic structure of protein. It is basically a sequence of amino acid which is positioned from left end to right end. So the left hand will include the N terminal, right, which is the amino group, and the right end will have the acid terminal, that is the C terminal. Let us see what is secondary structure. So here the protein thread is folded in form of a helix, as you can see here. Only right-handed helices are found here. They can be alpha helices or they can be beta plated sheet. Examples would be keratin or silk fibers. Now let us look at the tertiary structure. So tertiary structure is basically a long protein chain that is folded upon itself like a hollow woolen ball. Here if you see, we see a 3D view of the protein. It is necessary for many biological activities of the protein. For example, myoglobin. Now, let us see about the quaternary structure. So, in quaternary structure, we see assembly of more than one polypeptide units, or these are called as subunits. You can see here, these are subunits, alpha alpha and beta beta. So, hemoglobin, for example, will have 4 subunits, 2 alphas and 2 beta subunit, and then the heme group will be present here. So this is what the protein structure are like. First we have primary structure, which is basically amino acids joining together. Then we have secondary structures, which has alpha helices and beta sheets, which has hydrogen bonding, which makes the protein strong. Then we have tertiary structure, that is when the amino chain folds over itself in a manner which resembles a ball of wool, right? There are various bonds which are present in tertiary structure to keep the protein structure together. Then we have quaternary structure in which we see subunits. So here the chains form a single protein by various subcomponents coming in together. Hemoglobin, as we learned, is an example of the quaternary structure. Now let us look at polysaccharides. So as the name tells you, poly means many, and saccharides means sugar, right? So they are lots and lots of monosaccharides combining together, polymers of sugar. These include starch, cellulose, glycogen, inulin. Starch is a homopolymer of glucose. That means it is made of various glucose molecules joining together. Cellulose is also a homopolymer of glucose. Glycogen is also a homopolymer of glucose. But inulin is a homopolymer of fructose. We see various complex polysaccharides formed of amino sugar like glucosamine, acetyl glucosamine. Chitin is a homopolymer of N-acetyl glucosamine, which is seen in exoskeleton of arthropods. Starch, as we all know, is the structural polysaccharide, right? And cellulose is found in the cell wall of plants. In polysaccharides, you will see glycosidic bond. So what is this glycosidic bond? Glycosidic bond is basically a bond formed between individual monosaccharides. They are linked together. The two carbons are linked together by dehydration reaction. So you can see here glycogen is a polysaccharide. Glycogen is mainly found in the humans. You might have noticed for starch test, we always use iodine. This is the standard test for starch, right? When we add iodine on starch, then we see this dark blue or black color which is formed. So why is this? This is because starch forms helical secondary structure which holds the iodine molecules in a helical portion, giving the dark blue color that you actually see. For cellulose, this test is not relevant because cellulose does not have these complex helices and therefore it cannot hold the iodine together. So let us look at various polysaccharides that we have studied. Cellulose is found in plants. The subunit is beta glucose. It shows 1 to 4 bonds. There are no branches here. Amylose and amylopectin are different compounds present in starch. They are found in plants. Here the bonding is alpha glucose. 1,4 and 1,6 is seen in amylopectin. It has subunits. About 20 subunits is present here. Then we've got glycogen. Glycogen, as I told you, is an animal polysaccharide. Alpha glucose is the subunit. The bonding is 1,4 and 1,6, and it does have branches per 10 subunits. Now let us study about nucleic acids. So the nucleic acids are DNA and RNA. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. So as we saw earlier, nucleic acid is actually a heteropolymer of nucleotide. That is, many nucleotides are linked together to form a polynucleotide. A nucleotide will include a sugar, a nitrogen base, that is ATGC, adenine, guanine, thymine or cytosine, and a phosphate group coming together. This secondary structure of the DNA was first found by Watson and Crick. These are the scientists who found the structure of DNA, right? So they determined that the DNA consists of two polynucleotide strands which are arranged anti-parallelly and they form a double helix. The length of one full turn is 34. So there are 3.4 for each step, right? At each step, the strand turns to 36 degree. So there is 360 degree for a full turn. So as we saw earlier, the nitrogen bases, deoxyribose sugar and the phosphate group is what form the nucleotide, right? The backbone of the DNA strand is formed by these three things. The steps are formed by the nitrogen base, that is A, T, G or C, adenine, guanine, thymine or cytosine. Uracil is absent in DNA. In RNA, it will be. Now, one thing if you note here is that A always pairs with T with a double bond, adenine always bonds with thymine, and guanine always bonds with cytosine with 3 bonds of hydrogen, right? So, a phosphate molecule links the 3 carbon atom of one sugar of one nucleotide to the 5' carbon atom of the sugar of the succeeding nucleotide. There is ester bond between phosphate and OH group of the sugar. As there is one such ester bond on either side, it is called a phosphodiester bond. The bond between the sugar and the nitrogen base is called the glycosidic bonding. I hope you all have understood the structures of all the biomolecules so far. We will continue this chapter in the next video. Thank you.